Okay, guys, let's get started. So, just the two of you here currently. I'll introduce myself first. So, my name is Shubho. I'm currently a second year PhD student at ISC Bangalore. I primarily work with mostly genomic data, and my primary area of work is structural variants in different kind of genomics data. So today I'll be teaching introduction to dynamical models in biology. So I am the PMRFTA for this course, and I'll be taking these 2 hour sessions starting this from today. So 4 to 6 p.m. every Saturday for the next 3 Saturdays, including this one 4 Saturdays. I'll take this live sessions uh, uh, on the NPTEL course. So I'll urge all you to join the live sessions directly. Also, these sessions will be uh, recorded and posted on YouTube. So if you miss this, you can always go back and check the videos. But I'll strongly suggest to join these live sessions. Okay. So before moving on, I just thought I'll give you my introduction. And if you are okay with it, I'd like you to kind of unmute yourself and just tell me a little bit about yourself, your name, your background, so that I can get a better understanding of who I am teaching to, so I can tailor me my content according to that. So if you're all right, I would like to know your names and backgrounds, please.
Uh,
Yeah, yeah,
Sir,
Shambhu.
I am Sombhujana. I am recently completed my graduation in microbiology from Midnapur College Autonomous.
Okay,
Now I am now I am get admission in Pondicherry University in bioinformatics.
okay, okay.
Uh,
That's good. That's good. Okay. So, do you have any mathematics background in your bachelor studies, or do you remember calculus and other basic mathematics from class 12, or uh, how are you comfortable
no,
with?
sir. I have no graduation level. I have no mathematics, but 10+2 I have mathematics, and I remember the calculus, basic calculus, and derivative
Okay, okay,
calculus.
that's good. That's good. Okay. So, I one thing before we start. So, sir has also instructed me uh, to keep in mind that maybe many of you are not from mathematical background. So, I'll try to keep the math part as simple as possible. If you face any issues, you're always welcome to ask me in this live sessions. In the end of the live session, also, I'll provide you with my email address. If you find anything difficult to understand from this course, you can always email me as well if you want in your free time. I'll reply whenever I can. So yeah, I'll keep this in mind. Not everyone is from similar background, so bear with me over the course. Okay. Yeah. So Jairam, you want to say?
Hi, this is Jairam Prakash.
Yeah.
I'm second year PhD scholar
Okay.
in mathematics
Okay.
from Coimbatore.
Okay.
My field is fractional differential equations.
Okay. Okay. I'm just curious. Why are you taking this course? Like, what is the objective?
Because I'm more interested on biological models.
Okay.
So this course will help me to study the models and behaviors, dynamical structures, everything.
Okay. 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 Got it. Uh, uh, Aishwarya, would you like to unmute and tell us a little bit about yourself and your background?
Hello, I am Revati,
Okay.
and I've graduated biotechnology, BTech biotechnology,
Okay.
and I took this course just out of curiosity, I guess, and I wanted to learn a bit about mathematical modeling because I thought for my higher studies I can take a related course.
Okay. Okay. That's good. That's good. Okay. Now we have got to know everyone, just three of you here, but still. let's get forward with it. I'll share my screen. Can you see the PPT?
Yes.
Yeah, okay. So, yeah, hello everyone again. This is the live session for week 1 of the NPTEL course Introduction to Dynamical Models in Biology. My name is Shomo, and I'm a PhD student at IISc Bangalore, and the course instructor is Professor Biplab Ghosh of IIT Guwahati. So, in general, in this live sessions, I'll first cover a little bit about a little bit of recap about what Sir has taught in this particular week, and in the end, I will do some hands-on problems and solve some questions in the end. Okay, so let's get started. So first, why use mathematical models in the first place? So a lot of you are curious about mathematical models in biology, but in general, why use mathematical models in the first place? So the major advantages of it is basically it provides us with a it's a very highly precise language, and it provides us with a platform to formulate our ideas and kind of simulate the underlying assumptions that we make about certain systems, and kind of get results in silico to have a better understanding of how the system is working in real life. So in the real life system, maybe you cannot manipulate all the stuff for example, in a biological system, it's very complicated. So understanding how each and every part of that system works is very difficult from an experimental point of view. You need to shut one down, then turn one on, then check if certain functions are getting hampered or not. In this way, in experimental biology, you kind of determine what are the different functions and what are the different carriers of different parts of the system of the biological system, be it a cell, be it a predator prey system, or whatever it is. So mathematical models gives you uh, an alternative way to find out what exactly is going on inside the system with the help of certain assumptions as we go along with the way. Just a second. Okay. Uh, and the mathematical models uh, have been useful to us for many centuries, to be precise. But recently, due to the leverage that we have of computational capabilities that we have currently, so it is possible to simulate huge models using utilizing the computational power. So the models are becoming more and more complex, and uh, we are able to simulate these with the help of the current infrastructures that we have, and it is getting better each and every day, and so is our models. So uh, how are mathematical models used in biology? So understanding complex biological systems require more than just characterizing individual molecules. It involves comprehending other stuff like molecular pathways, interactions, how different kind of genes and proteins and pathways are regulated. Just a second. And they provide crucial role in investigating how regulatory processes are interconnected and how these disruptions of particular processes can uh, contribute to a disease. So and these models helps us systematically analyzing system perturbations, guiding experimental design, and evaluating molecules as therapeutic targets. So as I was saying, biological systems in general are very complicated, so it is very hard to understand uh, what exactly is the role of a particular component of that system, be it a gene, be it a pathway. So what we do is we try to formulate the functions of those particular elements inside the system using mathematical models, and we try to simulate them and try to perturb them to see what kind of results can we get if we say perturb a particular pathway, or we shut down a molecule that is producing a particular protein. So these kind of experiments, that is actually, if you want to do it in the wet lab, which will be very difficult to do, can be easily done using uh, can be easily analyzed using mathematical models. But one thing to keep in mind always, all the models that you are using are in some way or form wrong in certain aspects, because we are using certain assumptions to make those model, and in real life inside the biological system, those assumptions may not hold, and in most cases it does not hold. But why we make those assumptions is because it gives us an approximate idea about how the system will behave if we have those assumptions. But without those assumptions, we cannot even simulate those models, because those will become very, very, very complicated. And so we try to simplify those models using the assumptions. But in real life, not always those assumptions hold on. So there is some error associated with these models always. So that is a very important thing to keep in mind. And mathematical model is essential for biologists to analyze physiological processes, and for pharmaceutical industry as well, to support drug discovery and development. And recent technologies have brought us about a lot of data from different aspects. So omics is one of those technologies that we have used to uh, to get a lot of genomic data, a lot of transcriptomic data, even proteomics data, and we kind of we learn from those data, and we refine these models much better. And with the help of those data, we can make much more accurate mathematical models. Uh, so, just a second. Yeah. So, as I was saying, biological systems with numerous molecular components exhibit emergent properties, and these nonlinear reaction kinetics and complexities like cell structure contribute to this unpredictable behavior. Mathematical models help us capture these complexities, allowing in silico simulations. And these dry experiments, which are the simulations, are actually very cost effective if you compare it to an experimental setup, wet lab setup. So what we do is kind of we if we have a certain idea in our mind, maybe we want to check if that particular pathway is involved in a disease or not. So first, what we'll try to do is simulate that particular assumption of ours in a mathematical model. If we get encouraging results, we try to replicate that in a wet lab system. But what these in silico simulations of mathematical models gives us is a better understanding of about how the system is working in real life, and it narrows down us to a certain number of experiments and certain type of experiments that we can do on in the wet lab to get our results. So it kind of uh, the dry lab experiments and the wet lab experiments, the mathematical model simulations and the wet lab experiments, they kind of go hand in hand uh, in laboratories, where you can try to narrow down your your possible number and type of experiments with the help of mathematical models. This is highly cost effective, and it's actually a very good way of understanding how the system is looking. So yeah, the combination of dry and wet lab approaches in systems biology aids in hypothesis development. And in metabolic engineering as well, these kind of mathematical models are used. Will not go into deeper for metabolic models, but if there are there's a vast applications of mathematical models in metabolic engineering as well. So there, the models suggest genetic modifications that can be used, that can be incorporated inside a organism, so that we can get maximum productivity. Say, for example, you're generating, or you're producing insulin from an genetically engineered E. coli. So there you can use this kind of mathematical models to see what modifications can bring about the maximum production of insulin from the particular microbe. So questions like these can be answered using mathematical models, and which in turn can be proved using wet lab experiments. So we understood the part why we use mathematical models, specifically in biological systems. Next part is this are the different steps of using this particular type of modeling in biological systems. First, and the most important part of using mathematical modeling, is identifying the need, the, to define the purpose of the model and what it aims to address. So the first and the most important thing is to have the question you want to answer. You need to be very clear about what you are trying to answer, because your model building and simulation will revolve around that particular thing. So identifying the need is the most important thing. After that comes different caveats of the model. For example, parameter or variable listing, identifying key parameters and variables required for the model. Then you identify available relevant data which can help you kind of validate your assumptions of your results from the assumptions of your model, and identify circumstances and assumptions that can be applied as well. And after that, this is another very important step in the modeling part, is governing physical principles. So understanding what is actually going on in the particular scenario, inside the particular system, is very important to create your models. So identifying physical principles, like for example chemical kinetics, or a particular type of interaction that is happening between two molecules, or it may be anything, but we need to have a clear idea about what are the governing physical principles that is going on inside the system, and what we are modeling using the mathematical models. And then after that, once you've identified these, you can write down the equations, do the calculations, and have a set of results. And then you try to check for consistency of this model using real life data, and kind of validating your model. So basic steps are listed down here, but the most important part to remember is to have a very meaningful question you want to answer from the model. If your question is not clear, your model will not work properly, because everything around your model will revolve around the fact about the what question you are trying to answer. We'll come to some examples later, but I just want to tell you this now on. Okay, so moving on. So uh, governing physical principles, what we mean by that. So for example, one of the governing physical principles inside the cell that is always there is the law of mass action. This is one of the oldest laws that is still very relevant. This law is more than even 150 years old, but still relevant to us. So it's a very good it's a law that has stood the test of time. So if you consider a simple chemical reaction like this, A and B giving you C and D with the coefficients small a, small b, small c, and small d, and according to the law of mass action, the rate of the rate of this reaction is expressed as K with your product your substrates A, concentration of A and concentration of B raised to the power of their coefficients, where these are concentrations where if you put A and B in square brackets, these denote concentrations of this particular reactants, and your small a and small b are showing your stoichiometric coefficients. Your K here is the rate constant, which is a positive parameter that depends upon temperature and other conditions. So this is one of the uh, very widely used equation in mathematical modeling, while we are using mathematical modeling for say particular trying to model particular events that is occurring inside the cell. Say, for example, some kind of enzyme kinetics or some kind of metabolic reaction that is going on inside the cell. So this is law of mass action comes in very handy to kind of quantify these different kinds of reactions inside the cell using the concentrations of the substrates present. So this is, say for example, I was saying in enzyme kinetics, a law of mass action can be applied. So this is a simple reaction many of you might have seen. So E is your enzyme, S is your substrate. K1 and K minus 1 are basically the rates of the opposite reactions. So K1 is the rate of the reaction of forming ES from E and S, and K minus 1 is the opposite, formation of E plus S from ES complex. So the ES is basically the enzyme substrate complex, which basically breaks down to give you the enzyme back and the product. So the enzyme is not used up during the process, which we know enzymes are catalysts. Basically, they do not get used inside the process, but your substrate is getting converted to a product. So two steps are there. One is formation of the enzyme substrate complex, and second step is breaking down of the enzyme substrate complex into uh, enzyme and product. So I'll give you a couple of minutes if you can write down the equations for the enzyme substrate complex and the product and the substrate mean. Then we can move on. I'll give you a minute if you write it down or if you want to think about it. Sorry. Just a second, guys. Yeah. Sorry. Yeah. Okay. So this is how the different this is how the rate of formation of product or rate of using up of substrate or the rate of formation of the enzyme substrate complex is given by the law of mass action. So it is very intuitive since when you are uh, checking the using up of the substrate, it is negatively dependent on this E and S concentration and positively dependent on the formation of the ES. And uh, they are given by they are multiplied by the rate constants. Similar is the case for the rate of using up of ES, which basically is getting formed from E and S. So a positive sign here, a negative sign here because ES is getting broken down to E and P by K2, and ES is also getting broken down to E and S by K of -1. So both of the parameters, rate parameters, are also included in this equation. And the rate of formation of the product is quite simple. It's just K2 into the concentration of the enzyme substrate complex. So just to keep in mind, the second equation, uh, the rate of change of in the enzyme substrate complex, it depends both on the formation of the product, and also there is an equilibrium here where the ESI is also getting broken down into the enzyme and the substrate. So both of the rates you have to consider as it is done here. So I hope all of you got that right. If not, it's quite simple. Just try to understand from where it is coming, from where the products are getting formed. If the particular thing is getting used up for the formation of that product, there will be a negative sign. If the particular thing is getting formed during formation of that product, it will be a positive sign. So it's quite intuitive. So just wanted to give you a overall idea about how we use different models and uh, different kinds of wet lab experiments, how they are designed across in in accordance with the mathematical model. So you have a biological phenomena. You'll have some experimental data from that. From the data, you'll find certain patterns and certain governing principles, and you apply mathematical models to that. Now, from the mathematical model, you'll have from the result, you'll have an hypothesis. You check out that hypothesis using wet lab experiments if that hypothesis is good enough. If not, you do another round of computer simulation with the help of new data, and you refine your mathematical model again, and then you again get the hypothesis. So you kind of trying to refine your hypothesis using new data and tweaking the parameters of the model and checking how your models are differing from each other. And you, after you have gotten a solid hypothesis, you test it out on your wet lab experiments, and you get a new set of data from the wet lab experiments, and you incorporate that data inside your mathematical model to try refining again. So it's kind of a a positive feedback loop. You have a model. From that model, you are getting a hypothesis, and using the hypothesis, you are uh, you are constructing a wet lab experiment, and from the wet lab experiment you're again getting a data which can be used to further refine the mathematical model. So it's goes on like a positive feedback loop. So this the in silico or the dry lab computer simulations including mathematical models goes very well hand in hand with the wet lab experiments. So if you're a good researcher, you'll have both of them will be very important to you, because uh, the insights you get from your mathematical models eh, actually saves you a lot of money and a lot of time and resources to narrow down your experiments to be done in wet lab to kind of support your hypothesis. So using both is very important. So now we come to the part where we can see different kind of biological modules as ODEs. So, for example, if you consider a concentration of any substrate, that can be an mRNA, protein, small molecule, a metabolite, or any kind of reagent, and uh, you want to check the concentration of those particular the change in concentration of those particular substrates after a small amount of time. This is basically the expanded Taylor series for dxdt. So x of t plus delta t is given by x of t plus k into xt of and delta t. K is your basically the slope of the of the curve at that particular point. So if you take delta t tends to zero, if you take that limit, it basically uh, if you can imagine the delta t is basically very small. So the next point t plus delta t is very close to t. So that is what is denoted by dx by dt. So what it is denoting is that the minute change in x due to the minute change in t. So the smaller the t, the better is an estimate about how your x is changing across different time points. So this is how different kind of these differential equations are written for different biological models. For in this case, it's basically a reaction you can consider where a substrate is it's getting formed and the concentration of the substrate is given by x. And here the equation denotes dx dt equal to kx, meaning the rate of formation of that particular substrate is dependent on the concentration of that substrate only. So rate of formation of x is given by dx dt and which is proportional to x itself. So this is what the differential equation is telling you. So if you want to find the exact equation and you want to find the concentration of x at a particular time t, at any time t, what you need to do is you need to find the you need to integrate this equation to find the original equation of how x is dependent on t. So how x is actually represented by t. So if this is a very simple equation, if you differentiate this will come as a log scale or a e to the power t scale. So uh, I'll come to how to solve these equations a little bit later. I just wanted to give you an overview about how we think about differential equations in case of biological models. So as I was saying, this is a fairly simple equation. So this is a first order linear uh, autonomous ordinary differential equation. So if you leave the autonomous part out, the first order is the order is denoted by the highest derivative there. So here the highest derivative is only one time because it's dx dt. If it would have been d2x dt2, that would have been the order of 2. Linear because it is directly proportional to x, not x square. So the dependent variable, which is x here, that should be in a linear form, basically x to the power 1. The moment it exceeds 1, if it is 2 or 3, it does not remain linear anymore. It becomes non-linear. So uh, here x is your state variable. The k is a constant, which is given by a parameter. Uh, a very important thing to remember is it does not describe any elementary component of the process. It just describes the rate of the process. It describes the reaction rate. That is how the concentration of X varies with time. It is not describing any elementary component of the process. This is just a constant parameter that describes the rate. Okay, so moving on. So as I was saying, this is a fairly simple ODE. You just separate out the different variables on both sides. It is doable. So you take so this was the equation. You take x to the left, so dx by x equal to k dt. This is what becomes. Then you directly integrate this because you can separate out the variables on each side. So here the initial concentration of x at t equal to 0 is given by this x0, and so this integral is basically a finite integral a finite integral from x0 to xt. So x0 is the concentration of x at t equal to 0, and xt is the concentration of of x at time equal to t. And here on the right hand side, the integral is from 0 to t. So dx by x, this is a fairly simple integral. It comes out as log. So when you put in the the boundary values, the xt and the x nil, so it becomes ln xt by x naught equal to kt. You rearrange this a little bit, it becomes something like this. So, and if you exponentiate it, it comes out to be x naught e to the power kt. So it's an exponential curve. The concentration of x varies as an exponent of the time t. So it increases exponentially. This is what it means. So this k is very important because the value of k determines if it is exponential growth or exponential decay. If k is greater than 0, it grows like this. And if k is less than 0, it is actually showing an exponential decay. The concentration is decreasing. So the value of k is actually important to determine if the system is growing or it is decaying. So uh, these are how the curves would look like depending on the value of k. So the k is an important parameter. The value of uh, using different values of k can give you different kinds of curves. So k is an important parameter. Okay. So after this, the last ODE that you saw here, it was a fairly simple one. It's dx dt equal to kx. So this is an ODE where you can just cross multiplying, you can just separate out the two variables on the two sides, the dependent and the independent. But that is not always the case. So for example, this is an example of a very complex system. So this is a ODE model of a biochemical network. It consists of three proteins. Each hypothetical protein can be in one of two states, which is basically phosphorylated or unphosphorylated. And the network includes three interactions, as you can see here, three different kind of interactions with the curves with circles at the end representing activation of the respective reactions between different proteins. So this is basically the activation. Basically, C is activating the forward reaction from A to A phosphorylated. So uh, again, A is kind of accelerating the process of B phosphorylated turning into B. So these are coupled reactions. What are coupled reactions basically? So individual reactions uh, which are somehow involved in regulating other reactions. So for example, in the first reaction, A is getting converted to AP, but this whole step is getting regulated by another concentration, another substrate, which is C, which is involved in its reaction of its own. So this is what we call coupled reactions or basically coupled ODEs we need to write for describing this particular system of reactions. So uh, even with this simplified network containing three proteins, it is hard to predict based on intuition alone what the network behavior will like. So for example, if you say, if I increase the concentration of A, is the concentration of B phosphorylated going to increase or decrease? So here is A. Say, for example, you increased A. Your question is, will the concentration of B phosphorylated increase or decrease? Just by seeing at the equations alone, it is not very easy to say this. Okay, will it increase? Will it decrease? So here comes the point of the different mathematical models with ODEs with Michaelis-Menten expressions for all reactions. So that is the governing principle, physical principle here, the Michaelis-Menten expression. And this is how different kind of the different reactions are written down. The change in concentration of A, B, and C. As you notice, the change in concentration of A is dependent on C, and the change in concentration of B is dependent on A. So uh, the substrates are not acting alone. They are the change in concentration of the substrates. It's dependent on other substrates as well. So this is what a coupled ODE looks like. So this is a more complex system of equations than what we saw earlier. That was a fairly simple one, but this is a much more complex set of equations. So this is what are known as coupled ODEs. So see, these equations are much more complicated. You cannot directly separate out the variables and directly integrate them. So what we use are numerical methods to understand the behavior of these particular equations or reactions. So I'll come to numerical methods a little bit later, but these are the kind of places where you should use numerical methods, because analytically solving these, even if possible, first of all, it is highly likely that it is possible to solve these kind of equations analytically, because the real life differential equations that will come across for different models in systems, 90 or 95% of those equations will not be able to solve using analytical methods. You can try different tricks to solve it. Some of it will maybe be solved, but maximum, like over 90 to 95% of the equations, you will not be able to solve using analytical methods. You need to use numerical methods and see across time how the concentrations are changing. So this is the C panel is the result. Basically, how the different concentrations of A, B, and C are changing. And this is actually done using numerical method analysis using Euler's and Runge-Kutta order form method. So yeah, just wanted to give you an example about how complicated different systems can look like, and how these different kinds of substrates can be coupled together to form a whole system of equations. So yeah, moving on, that was another example. This is another example. So this is an example of the NF kappa beta kappa B signaling pathway. Many of you, I think, will be familiar with this signaling pathway, but I'll just go over it. So NF kappa B is a protein that plays a role in many important processes in the cell, such as inflammation, immunity, cell growth, and survival. It is normally found in the cytoplasm, but it must move to the nucleus to turn on genes. So it's basically a transcription factor which needs to go inside the nucleus to bind on the genes and facilitate the transcription. So to prevent NF kappa B from moving to the nucleus inappropriately, it is normally bound to inhibitory proteins called I kappa Bs. So these NF kappa beta Bs are basically floating around inside the cytoplasm, and like they can easily go inside the nucleus. But that going inside the nucleus and uh, regulating different genes inside the nucleus is uh, is controlled by another set of proteins called I kappa B. When a signal tells the cell to activate this NF kappa Bs, the I kappa Bs are degraded and the NF kappa B can move into the nucleus. So in its inactive state, NF kappa B is bound to I kappa B. But when it requires to move into the nucleus, this I kappa B is degraded and the NF kappa B moves into the nucleus. So one of the genes that NF kappa B turns on codes for a protein called I kappa B. So you see the irony here. NF kappa B is negatively regulated by I kappa B. When I kappa B is degraded, NF kappa B goes inside the nucleus to turn on I kappa B again. So it is kind of a very well regulated system. When your inhibitor is degraded off, you go inside the cell and you try to kind of do your function, but to check that your concentration doesn't exceed a particular limit, and that process is well regulated. So the I kappa B is I kappa B is a gene that is turned on by NF kappa B itself. So there is always a level of equilibrium between NF kappa B and I kappa B, which forms a negative loop. And the cycle of activation and inhibition helps these NF kappa B levels to keep in check. So how do you study this phenomena using mathematical models? So this is how it is. So as I said, NF kappa B is a complex regulatory network, and that is controlled by inhibitory factor I kappa B alpha. The cellular concentrations of the relevant molecular pairs over time can be expressed by formulas such as shown here. So this is basically I kappa K alpha and different concentrations of NF kappa B, and so this is a basically a complex set of ODEs that are used to kind of model this whole feedback loop and the regulation of NF kappa B and going inside the nucleus and how it can turn on genes, and also how it will be repressed after a certain concentration because I kappa B will be forming again. So this whole thing can be modeled using a set of differential equations. As you can see, this is another set of complex differential equations. You cannot solve them analytically using hand. What you need to do is uh, uh, use numerical methods to solve and simulate this model and check how the different kind of concentrations are changing. So this is another example of a complex biological system where you can use mathematical models and try to see how the behavior of the whole system across time. So if you are interested, there are a couple of papers which actually go through these in very detail. If you want them, please go through these papers. Hoffman et al. 2002 and Lipniaki et al. 2004. These are two papers which have extensively studied this particular process and using mathematical models. So okay. I'm sorry, I did not understand your question. So, can you explain what you're trying to ask? Okay, so the variables will depend on your model. So, if your model has only one kind of, say for example, your model has only uh, one substrate that is getting converted into a product. So, uh, the number of variables inside in your model will be one, and for naming of the variables you can use any name as long as it is consistent across the whole model, so that at the end when you try to get the results for that particular variable, you know what that variable is. But I, I'm not sure what you're asking, but you can name anything, but you should know what it is actually meaning. And the number of variables of your model will depend upon the number of things you are trying to model. If you're trying to model the decay of the substrate that is inside your system, that will be one variable. If you're trying to model the formation of the product in your system, that will be another variable. So the number of variables will depend about what you are trying to answer. But yeah, I you can name anything as long as you know what it means. Okay, so moving on. So what are the different general assumptions that we make about ODE-based models? So just remember one thing: whenever you are using mathematical models in any field, there will be a set of assumptions associated with the model, because if you want to exactly mimic a real system in terms of mathematical model, it becomes very, very, very complicated, and you will not be able to solve the model, and there is no point of having that model. So always with a model, be it in any field, specifically biological systems, will have a set of assumptions. So what kind of assumptions we have? For example, the system is homogeneous. So why is it important? Why do we assume that the system is homogeneous? Say for example, you are talking about a particular reaction that is going on inside the nucleus, or uh, in, inside the nucleus of the cell. Say for example, you're talking about the different transcription factor that is going inside the nucleus and trying to regulate a particular expression of a gene. So why do we assume that the system is homogeneous in this case? Because in real life it is not the case. In real life what happens is the concentration of that particular substrate is much more inside the nucleus than the whole cytoplasm. So there is always a concentration difference depending on the function of that particular substrate. So in real life, there will always be a different concentration across different gradients of the cell. So if you move from cytoplasm to the nucleus, maybe the concentration will increase. If you move from nucleus to a particular gene of the in the DNA, the particular area of the DNA, there may the substrate concentration may increase more. So the concentration is not homogeneous actually inside the cell. But if we want to consider this, what happens is we need to have separate models for separate areas of the cell. So a model for the model for the cytoplasm of the cell or for that substrate, a model for the nucleus of the cell for the substrate, a model where it is regulating the gene for the substrate. So what it becomes is, it becomes unnecessarily more complicated and more difficult to solve. So these kind of assumptions are what makes the mathematical models much more simpler. Uh, Given that these assumptions are not always true, obviously these are not true, but these are very good approximations of what we can see inside the cell. So uh, these assumptions are very important while making mathematical models, because without these assumptions we'll be lost. So uh, the first assumption is the system is homogeneous. The second assumption is all cells in an ensemble behave similarly. So that is also something. So when we are modeling a system of a tissue inside a particular organ, we assume that all the cells inside the tissue are the same, which is not the case in real life. There will always be some heterogeneity inside the tissue. Some cells will have a different kind of gene expression. The other cells will have a different kind of signature of gene expression. So it is not true that the system will have all the cells will behave similarly, because all the cells may not be of the same cell type. But if you want to factor all this in, becomes much more complicated. The models assume that reactions and processes occur instantaneously compared to the time scale of the overall dynamics. So that is also a thing. In real life, in real biological systems, there are time scales of particular reactions. So some reactions happen in milliseconds, some reactions happen across days. So this is also something that is varying across different functionalities of the cell. So some functions can happen in just like this, like in a millisecond. Some functions can take certain days to happen. So there is a variability inside the cell about different kind of functionalities it performs. But during using of mathematical models, we assume that the reactions and the processes occur instantaneously. Other assumptions include that the parameters such as the reaction rates, the diffusion coefficients, or any other kind of parameters that we are using, that they remain constant over the time period the reaction is happening. Or else there will be, which is actually, if you think about it, it's not the case in real life, because it cannot, the reaction rate will not be constant, because it will depend on basically the environment of the cell. So in one part it may be constant, in one part it may increase, in one part it may decrease, depending upon the regulation and the systems inside the cell. It is not constant if in the real system, but we assume it to be constant because it otherwise it unnecessarily complicates the model. So yeah, this is a general set of assumptions that we have for different kind of models, ODE based models that we use in biology. But to keep one thing in mind, your assumptions will be based upon the model you are trying to make. So not all the models will have same assumptions. The assumptions will depend upon the question you are trying to answer. So that is very important. Just keep this in mind. Like you will not have a set of assumptions that is same across all different kind of models you're using. Your assumptions will be based on the model you are trying to make, the question you are trying to answer. Okay. And so this was just an overview of the different kind of assumptions that we make. So moving on. So a little bit more about differential equations. So the order of the differential equations I just told a little bit earlier is basically the order of the highest order derivatives. So for example, if you're the highest number of derivatives is basically the highest number of times a particular dependent variable has been a derivative. So for example, in this case, dy dx equal to e to the power x, the derivative has occurred only one times. Y has been differentiated only one times. So the order of this equation is first order. In case of the second equation, y has been differentiated here four times. which is basically d4 y dx4, which means y has been differentiated 4 times with respect to x. Yet y is the dependent variable, x is the independent variable. So the equation is a fourth order differential equation. So can anyone write in the chat what is the order of third equation?
Three.
Yeah, it is 3. So just to be clear, sorry, sorry guys, my screen got stuck. Sorry. Yeah. So yeah, one thing to keep in mind, there is you see there is a square. It doesn't matter if it has a square or to the power 10 or to the power 15. What matters for the order of the equation is the highest number of times the dependent variable has been differentiated. Here the highest number of time the dependent variable has been differentiated is 3. So the order of the reaction is 3 in this case. uh, so just to keep these small stuff in mind, because questions might come in the exam in the form of MCQs and asking you what is the order of this equation, or is it a linear equation or not, or etc etc. So the small things about differential equations is it's good to know. So yeah, different kinds of ODs that can be there: first order, second order, nth order ODs, depending on the number of times the dependent variable has been differentiated maximum times. So another concept in OD is basically if it is linear or non-linear. A linear differential equation, uh, it's basically the coefficient of the dependent variable in all order derivatives of y are functions of t or constant terms only. So linear uh, OD will only be possible when the dependent variable has the uh, uh, highest coefficient of 1 basically, or is it's a function of the constant. So a linear ODE will not have basically dy dx equal to y squared is not a linear ODE. Dy dx equal to y will be a linear ODE, or dy dx equal to a constant k will be a linear ODE. But whenever the power of y becomes more than 1, it becomes a nonlinear ODE, depending on the power of y. So linearity and order are something that you'll come across many times inside this course, and also these kind of small questions can come in the exams as well. So keep your concepts brushed up for these small stuff regarding ODs. What is an order? What is the first order, second order? Different kinds of ODs. What if the OD is linear or non-linear? So these kind of concepts, please remember going forward as well. So yeah, another is a type of ODs that we find is basically with different kind of auxiliary conditions. You can have initial value problems where it's basically at the initial time your the value of your dependent variable is given, or it can happen is the value of dependent variable is given along a boundary. So say for example, at you know what is the value of the dependent variable at 2, you know what is the value of it at 4, and you're asked to solve the ODE and find uh, basically the area under this curve at 2 to 4 or something like that. So it can be possible that you are given certain initial conditions. Initially, what is the concentration of that particular substrate, or what is the basically at t equal to at when your independent variable is 0, what is the value of your dependent variable? At t equal to 0, what is the value of x? Or at x equal to 0, what is the value of y? So maximum problems that you'll encounter in this course and your exams as well will have initial value problems, but there are boundary value problems, ODs as well. I'll not go into much details of that, but I just want to give an idea that those exist as well. Okay, so moving on. So as I was telling, the numerical methods are not always, sorry, analytical methods are not always applicable, because the system of equations for that particular ODE will be very complicated, and you cannot directly separate out the variables and do a simple integration to find what the exact equation is. So in those cases, what we do is we use numerical methods. So one of the numerical methods that we use is basically Euler's method. So Euler's method is it's the most simplest form of numerical method available. So what we do is we have a h, which is the step size. We calculate the value of y for every step size of h. So the smaller the h, the smaller the step size, which is h, is better accurate y you get. So what we try to do is we know what is the initial value of y at say t equal to 0. What we try to do is try to increase t little bit by little bit. So for t equal to 0, we know y equal to 1. We try to find out what's the value of y when t equal to 0 plus zero point 0.5. Then we try to find what is the value of y when t equal to zero point 0.5 plus zero point 0.5. So by taking small steps in the x-axis, we try to find what is the value of y. So this is an approximation, obviously, so you do not get an exact curve of what y versus t looks like or x versus t looks like, but what you get is a very good approximation of what your curve might look like, or how your how your equation is looking like, and in general how your system is behaving. So you get a very good approximation. So remember this thing, it's not an exact process. Any kind of numerical method is not an exact process. What you get is a approximation of the solutions. So Euler methods is one of the initial of the first type of numerical methods that we learn, and one of the easiest of them all. But there are other complicated numerical methods as well, which is Sir has taught one, which is the Rangi-Kutta fourth order method. So it is much better at approximating the curve, which is basically the x versus t or the y versus t curve, much better than the Euler's method, because there they do not only take the once basically the step size is there as well, but what they do is basically they kind of try to have a better estimate of the slope using slopes of different tangents across the curve. So I'll just do one thing. I'll try to solve one of the problems uh, for Euler's method using one of the few equations using Euler's method. Before I do that, I just want to kind of discuss a few things before that. So just let me get my pen. Yeah. Okay. So before we move into Euler's method and different kind of numerical methods, I just want to touch on a few topics that Sir has also discussed. I just want to give another overview. So first is the uh, s- yeah, first is the spread of infectious diseases. So this is a model that Sir has already discussed, but I'll discuss it from my end as well. So what we assume here is basically simple, sorry, uh, it's a simple infection model. So before we move on to the model, what are the assumptions that we make? So the first assumption. So see, here we are making the assumption, as I was telling earlier as well, we are making the assumption based on the question we are trying to answer. Here the question we are trying to answer is basically what will be if you have an initial fraction of population infected, what will be the total number of people or the fraction of population infected at time equal to t. So at t equal to 0, you are starting with a fraction of population infected. You want to find out at t equal to t what will be the fraction or the number of people infected. So this is the question we are trying to answer. According to this question, we'll make a certain assumptions to simplify our model. The first assumption is that the total population is very large. So this is an assumption that we make because this is also something, this another form of this assumption is kind of assuming that this inside the system we are trying to model, it is very homogeneous. So what we are trying to say is the population is very large. It doesn't matter like uh, where and when the people are getting infected. So the only way the people can get infected are basically is that the disease will only spread when they are in contact with each other. So it is not that kind of a disease. So for example, eh, if you remember COVID, there was a time when people were very worried because surfaces, basic surfaces that we have in our daily life, say for example, tabletops or other kind of surfaces, coronavirus used to be kind of pathogenic in those surfaces as well. So if you kind of touch the surface after an infected person has touched that surface and some COVID-19 is there, and if you touch the surface, it can come to you. It is kind of transferable from surface to surface as well. So that is how a real life infection works, but we cannot model that if we want to keep our model simple. So this is another assumption that we make, that the disease will spread only when a person is coming in contact with another person. There cannot be a third way, there cannot be another way of passing on this infection, maybe using maybe by getting infected from different surfaces and other kind of stuff. So we do not assume that those can happen. We assume that strictly the disease can spread only when in contact. And the third major assumption that we have is that everyone can come in contact with everyone. So basically what we are saying is everyone is vulnerable. There is no quarantine. There is nothing that prevents a person from transferring the virus from himself to an uninfected person if they come in contact. Anyone and everyone can come in contact. There is no concept of vaccination, or there is no concept of quarantine or anything like that. So everyone has an equal probability to get the disease from or I should say, every uninfected person has an equal probability of getting the disease from an infected person. This is also a very important assumption that we make in our model. And other assumptions include that the population remains constant. So basically what we assume is there is no death or birth inside in the population when when the disease is getting spread. So basically, no people are dying or getting no people are gi- basically giving birth to other people, because if we do that, we have to take into consideration the birth rate and the death rate of the population as well. So to keep our model simple, we also assume that the population remains constant. So which basically means is no death or birth. And the final and another very important assumption that we make is that no one gets cured. So what can happen is, like, say for example, an uninfected person is got infected from another person, but that person will remain infected for the rest of the time period that we are interested in. We do not assume that person can get better and that person can become uninfected after a certain point of time. So what we assume is once you're infected, you are infected for the whole period of time that we are looking for in the model. So this is also another very important assumption that we make. Now, if we get to the model, so if you assume that x is the fraction of population infected, and obviously if x is the fraction of population infected, 1 minus x will be fraction of uninfected population. Then the rate of change of infected population is proportional to the fraction of infected population and is also proportional to the fraction of uninfected population. So your dx dt will depend on both x and 1 minus x because sorry, I think somebody is trying to, yeah.
Hello.
Yeah.
Why you have taken 1 minus x?
So if you're an infected person, the number of people you can infect is 1 minus x. So the rate of change of infected person is also dependent on the number of uninfected person. If you think, is it not? So you have a set of persons who are infected. How many persons can they infect again? Which is that dependent on? That is dependent on the number of uninfected persons, which is basically 1 minus x. So your change in the number of infected person is dependent on both the number of infected persons and also dependent on the number of uninfected persons as well. So it's a proportionality that we are using. The change of the infected person is proportional to the product of infected and uninfected.
Okay.
Okay, so basically we are trying to see which basically change in a particular so we have two different groups. We have the uninfected group and the infected group. We want to check how is the change in the infected group dependent on others. So the change in the infected group will depend on the number of people in the infected group. It will also depend the number of people in the uninfected group because those are the people they can infect. So it is dependent on both, and we take it as a proportionality equation. So after this, what we do is we introduce a rate constant. So it becomes dx dt equal to r. R is your rate constant here, x into 1 minus x. So this is an equation sir has already shown you how to solve, but I'll just quickly go over it. So this is a fairly simple equation in the terms that you can just separate out the variables. So x is your dependent variable. So you on the left you separate out your dependent variable, and on the right you separate out your independent variable. R is a constant, doesn't matter where it is. So on the right side is your independent variable dt, and on the left side is your dependent variable dx. Okay, so if you So I'll not solve this again because sir has already shown you in detail how to solve it. But what it comes down to, if I remember correctly, is x. Just a second. Okay, well, let's do it all over again. Huh. Okay. So what you do is you integrate. So if your initial concentration is x naught, the final concentration is x. So this becomes dx by x into 1 minus x equal to r dt, and here you integrate from 0 to t. So at t equal to 0 you assume that your initial concentration is x naught, and t equal to t you assume your concentration is x. So you do a simple integration of this. So what it becomes is First you have to separate out. So x naught to x. So this becomes 1 minus x plus 1 by 1 minus x dx equal to rt, because it's t minus 0, so I'm directly putting as rt. So if you do this integration, so what happens is log of x minus log of 1 minus x, x naught to x equal to rt. So I'm assuming that you guys know that if you integrate 1 by x, it comes as log x. And why this negative sign is coming? Because the sign of before x for this 1 minus x is negative. So what you do is you the actual integral will look like log of 1 minus x, and also you have to differentiate this term as well because it's not exactly x. So when you differentiate 1 minus x, you get minus 1. So here the minus sign comes up. So that is the reason for the minus sign. And then if you rearrange this, you'll uh, get x by x 1 minus x naught x equal to rt. Now you put the value of the initial concentration, you put the value of the you want to find the value of x, you put the value of initial concentration here, you will know the value of r, you will know the value of t. If you put all these variables here, you will get the value of x. So this is how you kind of solve differential equations. This is an analytical method that we have used to solve here. But one thing to keep in mind, this you only were able to do because you were able to separate out the variables here. So x on one side, t on one side. If this would have not been possible, you will not be able to do it so simply. There are other techniques to analytical methods that you can use to solve a little bit more complex equations, but those are also limited to a certain extent. So after that, you have to go and use numerical methods, or else like a lot of your time will just go to solve these differential equations, which may not be able to solve using analytical method as well. So overall it is advisable if it is a complex equation of any sort, use numerical methods. It's much easier, and anyway you want to know the approximate values as only because your model has inherent assumptions. So what, even if you get an actual value from here, say for example here you are given an x naught and everything, you're given a r and a t, but the value of x that you get is considering the number of assumptions that you have made already. So it's actually not a very accurate value anyway. So there is no issue kind of approximating the value a little bit further. So that is the idea, that it is better to use numerical methods in cases where the the equations are too complex or too long, or like you do not want to solve it like that. Okay. Uh, so this was one. Let me draw another one. Okay. So I'll solve one Euler method sum towards the end. Before that, I want to go through a few questions um, that sir has actually asked me to discuss with you. So first, let's do this. Say, for example, this is a model. This is A, by the way. So this is a graphical model. So here, X is getting converted to Y. A and Q, can you tell me what are their properties? What is the representation here? What does this A and arrow mean, and what does this Q and arrow mean? Can someone tell me? Right in the chat box.
A is for activating, and I think Q is inhibiting the reaction.
Yeah. So for example, in this one, so this arrow, what it means is she's completely correct. So this headed arrow is activation, what how we denote activation, and this T, or like the arrow looking like a T, is what repression means. So if you are given an equation something like this, say for example, x y, and this, say only the A is there, Q is not there. Can you tell me how many e- differential equations are required to characterize this function fully? So the question is, how many differential equations do you need to to get the information of this reaction in terms of a mathematical model? How many reactions do you need? Is it one? Is it two? Is it three?
Is it two?
Yeah, it's technically it is two. So for example, if you're concerned about the change in y, so you'll be denoting dy dt. If you are concerned about x, you'll be denoting dx dt. But if you consider carefully, dy dt and dx dt are basically negative of each other, nothing else, because the rate of concentration of rate of change of concentration of y will be negatively related to the rate of change of concentration of x, and vice versa. So these two equations are kind of, it's the same, dy dt and minus dx dt. Dy dt is the rate of formation of y, dx dt is the minus dx dt is the rate of usage of x, which are both basically same, because it's x giving y. Now, why is a not involved? uh, just a second, somebody's trying to, I think, enter. Why is a not involved? Because a, as you can imagine, is an enzyme. And if you recall, enzymes do not get used up in the process of the reaction. So dA dt doesn't make any sense, because if you are interested in a time period from the start to the end of the reaction, the concentration of A is going to remain the same, because it's an enzyme. It does not get used up. So to characterize this equation, this whole equation, you actually need either this or this, because essentially both are the same. So technically you need two equations, but both are opposite of each other, so you can make do with one. So this whole equation can be modeled, a whole reaction can be modeled using any one of these equations. And A is not required, because A will always remain constant during the time period. Like if you compare the initial and after the completion of the reaction, the concentrations of A will remain the same, because A is an enzyme. That's the biochemical property of an enzyme. Okay. Just a second, guys. Okay. So just to brush up, in these two equations, can you tell me what are the dependent and the independent variables? Say, for example, this is the equation. What is y? What is t? Which of them are dependent and independent?
Y is dependent, T is independent.
Correct. So these are like very basic stuff. I'm just trying to brush up certain stuff, certain basic mathematical things. And you're completely correct. T is independent and y is dependent here. So, and can you tell me what kind of equation is this? Is this what is the order of this equation? And what is it a linear or a non-linear equation? First, what is the order of this equation?
1.
It is 1, correct. And is it linear or non-linear? Please explain.
For that, we need a full differential equation. I mean,
Okay, but say for example, is it linear or non-linear?
linear.
Yeah, because linear is for having coefficients 0 and 1. If it was a constant, in this case k, or if it is something like y, both are linear equations. But the moment it becomes a y square, it becomes a non-linear equation. So these equations are linear and these are of order 1. So yeah, I am sure you guys know this. You might be thinking like uh, I need not go through this, but just bear with me because not everyone in the course will have a similar mathematical background to all of you guys. So yeah, okay. Uh, so let's solve a question. So for example, the I don't remember the question number. I'm just putting like this here. Okay. So for example, you are modeling the growth of yeast, and it is given by the following equation. So dp dt equal to r into p minus d into p. Okay, and here p is the population size, r is the rate constant. I'm putting initials, rc is the rate constant for growth. Sorry, d is the rate constant for death, and say assume p naught is your initial population. Now this is your equation, dp dt equal to r into p minus d into p. This is your equation, and p is your population at given time t, r is the rate constant for growth, d is the rate constant for decay, and you are given the initial population as p naught. So you are asked the question, what will be the size of the population at t equal to 50? So how do we do this? It's a fairly simple sum. So basically you're given the model in terms of a differential equation. Your rate of change of population depends on your the growth of the population and also the decay of the population. This is the equation given to you. You want to find what is the exact population of this system of yeast at a particular time t. So what you do, you want to try to find the exact relationship between p and t. So you want to find an equation which relates your p, which is your dependent variable, and t, which is your independent variable. So from this, it's fairly simple. You just need to integrate. So how do you integrate? dp dt equal to, if you take p common from here, it becomes p into r minus d. Okay, now you can easily separate it out. dp by p equal to r minus d into dt. Now you do your integration, p naught to p, t equal to 0, sorry, to t equal to, do this integration. It's fairly simple. There is a log on the left side. So it will become ln p by p naught equal to r minus d into t. So you have the value of p naught, you have the values of r and d because these are constants, and at time t equal to 50, you just replace 50 here. So 50 into r minus d. So this is what it becomes. Now if you want to further simplify it, you take an exponential. So p equal to p naught e to the power 50 into r minus d. Now you substitute the value of p naught, r and d, you get the population p at time t equal to 50. It's quite fairly simple. You have been given the equation. You just separated out the variables. You did a integration from the different values that you were given uh, for p naught, which is the initial population at t equal to 0. You know the initial conditions. So you put in the values and you get the equation. And after that, if you want to find the population at a particular time with a certain rate constant and decay constant, rate constant for growth and decay, and you have the initial population size, you can easily find out what the population p will be at t equal to 50. So yeah, this was also kind of a fairly simple one. So and so the last thing to cover for today is basically the Euler method. So I'm just putting it like this. Euler's method, which is basically numerical solution of ODE. So Sir has already explained this. I'll just go over it in brief. Uh, so for example, you have an equation like this. Dx dt equal to, sorry, I've written it again. Dx dt equal to 5t minus x. Now, if you see, this is an equation that looks fairly straightforward, but you cannot directly separate out the variables. So you cannot directly have x on one side and like x and dx in one side and t on the other side. So it's a little bit more complicated than it actually looks. So it is a little bit harder to integrate. So directly you cannot separate out the variables and integrate. And there are other methods that you can use, but I'm not going to that. We'll try to solve this using Euler's numerical method. So let's go to the definition of Euler's method. So Euler's method is given by dx dt, which is approximately delta x by delta t equal to, what it actually means is x at t plus delta t minus x at t. So that is what delta x means. So x at t plus delta t minus x of t. So the difference will be basically delta x, and delta t remains delta t. So this is how you define dx dt in terms of Taylor series approximation and Euler method as well. So now what you have is your dx dt is given as 5t minus x. So what you have is x of t plus delta t minus x of t by delta t, it's equal to 5t minus x. Okay. Now let us assume your initial given condition is at t equal to 0, x equal to 0. Now you want to find different values of x across different values of t. So I will not do many calculations, but let's do some. So let's make a table here, t and x. Okay. And so this will look for say 5 values or 4 values of t. So we start from 0. Our initial step size, which is the h here, it's basically this difference it's basically going to be 0.5. So we are going to move 0.5, we're going to move to 1, and then we are going to move to 1.5. Now we want to find the different values of x according to the values of t from using Euler's method. So we have come to this till now. So how do we move on from here? So at x equal to, sorry, at t equal to 0, what was our equation? Let's move here. X of t plus delta t minus x of delta equal to 5 minus x. So x of t plus delta t becomes, if you rearrange this, xt goes to the right and and multiply delta t to 5 times x. Okay. Now when t equal to 0, what is the value of x? So okay, so initially at t equal to 0, you're given the initial condition, the value of x is 0. Now you want to calculate the value of x when t equal to 0.5. You just substitute t equal to t equal to 0 and delta t equal to 0.5. So if you substitute t equal to 0 and delta t equal to 0.5, so this will be 0 plus 0.5 into x of 0 again. Again, x at time 0 is given as 0 and delta t is 0.5. So this whole part is 0 because this is 0, this is 0. You multiply 0.5 to it, it becomes 0, and x0 is already given as 0. So it is 0. So x of 0.5 is 0. So at t equal to 0.5, x is 0 again. So let's fill up this table. 0. Now we want to calculate for the value of x when t equal to 1. So same method we use. So here now we have the value of x of 0.5. So what we do, we take t as 0.5 and delta t as 0.5 again. Okay. So how does this look like? X of 0.5 plus, this will be 5 into 0.5. This will be 0 again because uh, yeah, x of 0.5 is 0. So this will be 0 here again. So minus 0 and again multiplied by delta t, which is 0.5 again. So again x of 0.5 is 0 here, 0 plus this is basically 2.5, and 2.5 into 0.05 will be 1.25. So the value of x at 1 becomes 1.25. You substitute in this table. Then similarly you do for 1.5. So uh, I'll not go through the calculation again, but I've done it before, so I'll just write it here. So 1.5 will be basically 1 plus 0.5, and then you write all these terms on the right here, and then you calculate these terms. What you get in the answer is basically 3.125. So this is how for different values of t you can calculate the value of x. So what after this, what you can do is have a plot of x versus t, and you can just plot this here. So at t equal to 0, x is 0, this is 1 point. At t equal to 0.5, let's assume this is 0.5, x is 0 again. So this is another point. At t equal to 1, so let's assume here x is 1.25, so it's here, somewhere here. So again, x equal to at equal to 1.5, x is 3.125, somewhere here. So your curve will look something like this. Now, the more values you calculate, say for example, x equal, what will be the value of x at t equal to 2? It will be somewhere t equal to 2 will be here, maybe x will be here. I'm just assuming here. So different at different values of t, you can get the different values of x, and then you can plot it to get an idea about how the function is looking like. So this is the crux of numerical methods, that you use different values of your independent variable to calculate the different values of your dependent variable based on this formula. It's a fairly straightforward formula uh, of differential equations, or you can say it's a, it's a simplified form of Taylor series, or it's an Euler method. So all of them, basically what it does is it you calculate the value of your dependent variable x based on the value of your independent variable by slightly increasing it. Here our increase was basically zero point 0.5, which is called the step, which is given by h. So our h here was 0 0.5. We increased the values of t by 0.5 to check how the x was increasing, and we got a curve like this. So this is in general how numerical methods work. So why I did this is because you need to be able to uh, solve these numerical differential equations using Euler's method by hand. Obviously, there are softwares, there are other softwares also, like in the next module, you'll see there are JSON, and then other softwares, then Excel can also do it. But doing it by hand is something that you'll require, because in the exams also questions like this will come. Not in a long format, but maybe in MCQs you'll be asked to maybe say for example, you'll be asked what is the value of x when t equal to 1. Then you'll need to solve it like this. So you need to have a practice of solving these ODEs using uh, Euler method. But just keep one thing in mind: Euler method is does a lot of approximation here. So this is very good when uh, the the equation is linear. For in this case, it's a linear equation. So the maximum power of x is 1 here. So this is a linear equation. So Euler method works very well for linear equations. The moment it becomes x squared or x cube or x to the power 4, the curve, uh, the approximate curve that we get from Euler's method, it's the deviation is higher than that we have for linear equations. So the ideal case scenario where you use Euler methods is for linear equations. So here also, I have mentioned the shorter the step size, the better is the approximation to the solution curve. And Euler method is considered inefficient because of large number of steps required to achieve a specified accuracy. Sorry. And the reason for poor accuracy is that Euler method approximation is best suited when yx is a polynomial of degree 1. The moment it becomes a square or a cube or a to the power 4, the errors try to errors tend to increase dramatically. So this is something that you need to keep in mind. And uh, to to handle this shortcomings of Euler's method, there is another method that Sir taught you as well, and which is very prevalent, which is the fourth order Runge-Kutta method. I'll not go into detail of this method. This is not this is, I know, is a little bit more complicated than Euler's method. So I'll not go into much of the details. You do not need to know exactly how everything is working here, but just remember this is basically sorry, this uh, the estimate of k is basically in Euler's method we are using the slope. Here it's a better estimate of the slope using multiple slopes at multiple points of time. So basically you are having slopes at delta t by 2, and basically not only t and delta t, you are also looking at the point of delta t by 2 and how the slopes are changing across these three points. So this, what you get is k something like this. I'll not go into the very details, but it is a much better estimator of the slope than just the slope itself. So in linear, why Euler is much more uh, good in linear equations is because in linear equations the slope is how your curve is uh, going to look like. But in case of polynomials, to the power 2 to the power 3, the slope is not exactly a very good estimator of how your next point is going to lie. So in that case, we need a better estimator, which is the Runge-Kutta fourth order. So this is just to say, this is not a make or break. If you don't understand it, it's fine. Just you need to understand that um, there are certain issues with Euler method. So that is the reason why we use Runge-Kutta, because if the polynomial is higher than one, Euler methods gives drastic errors, meaning like the errors are quite big and those are not very good for approximations. So there we use techniques like this fourth order Runge-Kutta method. Okay, so yeah, I'll just check. I think I have covered everything for today. I'll just check one thing. Yeah. So sir has also asked me to tell it to you. So uh, if you are not familiar with something like this, so this is what is known as the Desmos plotter. This is a free website that you can search in Google and find out. So here you can just simply write equations like this. It's fairly simple to write. Just you have functions and everything. You can just use the this platform to get to know how your equations and how your curves are looking. You can use these curves to have a better idea about how your equation is looking like. So say for example, you have a differential equation, you solve it, and you want to see how your curve is looking like, how your x is changing with t or your y is changing with t. You can use softwares like this. This is a free tool that is available online. This is known as the Desmos plotter. I'll just give the link in the chat box. You guys can use this and to basically see the different plots of different kinds of functions if you're not familiar with it. So this is something that you can do. And I think I have covered everything that I needed to for this week. But before we end this session, I just want to give a quick uh, overview of everything that we discussed. So we started with why we use mathematical models. Then we uh, studied how mathematical models can be used in biology. What are the different caveats? What are the things to keep in mind? What are the different kinds of assumptions that we need to use for different kind of models? And for modeling, the most important thing is to find the right question to answer and the right question to ask to the model, so that everything about your model revolves around that. Then we studied a little bit about law of mass action and how we can make ODE models based on law of mass action for different kind of biological systems, which includes enzyme kinetics. And after that, we saw how to solve ODEs, simple ODEs, couple ODEs, and a few examples of NF kappa beta signaling and a few coupled ODEs, how they really look in real life. And after that, we solved a few equations for ODEs, and we brushed up of what is an order of an ODE, what is linearity of an ODE, and finally how you can solve ODEs using Euler's method. So uh, that was all for today's session. If you guys have any questions, you can ask me, or if you have certain questions that you find after this session, I am putting my email ID in the chat box. You can always write to me. I'll find time and obviously try to reply to you. You can write to Sir as well. I think Sir is also very keen to uh, write this to you. So, Tony, I am not sure if I'm not supposed to give them directly to you. I'll provide them to NPTL. I'm not sure how NPTL provides it to the students, but I can take a look. But the whole lecture and the video will be uploaded on YouTube. You can always go and check it back. But I'm not sure if you can get the PPT or the note document. I'm not sure because I'm supposed to give it to the NPTEL people. So I'm not sure if they can give it to you. Okay. Yeah. Now you can always come back and watch these videos. This will be in my channel where I just type my whole name here if you want to find it later. So these videos, these 4 weeks ka videos, all will be in my channel. These 2-hour videos. You can, if you find anything that you want to go through, again, you can always go back to the channel. This is the name of my channel. You can go there and you can see the videos. Okay. Anything else? Okay, guys. So before I drop off, just to say, practice integration by hand, manually doing integration and differentiation, small sums, because for this course you should be able to do small integrations and differentiations manually. And also Euler's method, that's an important topic. There will be questions like from there in the assignments as well as in the final exam as well, where you need to uh, for do certain small sums and maybe put answers in MCQ format, but you'll be able to you'll be asked to solve sums like this. So my advice is practice these different types of ODEs, solving ODEs using analytical methods, using Euler's method as well, and and that's all. I hope you like the session. We'll meet next Saturday at 4 p.m. again. Okay. Thank you, guys.
Thank you, sir. Means a lot.
Yeah. Thank you, guys.